The human brain is arguably the most extraordinary and metabolically demanding organ in the entire known universe. Weighing just 3 pounds, roughly 2% of total body weight, it consumes a staggering 20% of the body's entire energy supply every single day. It fires approximately 86 billion neurons in a ceaseless electrical symphony of thought, memory, emotion, creativity, and consciousness. It processes 11 million bits of information every second. It rewires itself continuously throughout your entire lifetime in response to experience, learning, and yes, the food you eat. And among all the foods that science has studied for their impact on this magnificent organ, one ancient, humble, extraordinarily concentrated category of whole food stands out with remarkable and growing scientific consensus: nuts. From the walnut that uncannily resembles the brain it nourishes, to the almond revered since antiquity as food for thought, to the pistachio studied in cutting-edge neuroscience laboratories, nuts have emerged as one of the most powerful, accessible, and scientifically validated brain foods available to humanity. This script explores the complete story of how nuts affect the human brain, the mechanisms, the molecules, the clinical evidence, and the extraordinary implications for brain health across every stage of human life. Before exploring what nuts do for the brain, we must first understand what the brain desperately needs and why modern diets so consistently fail to provide it. The brain is composed of approximately 60% fat by dry weight, making it the fattiest organ in the human body. But not just any fat. The brain demands specific types of fatty acids, particularly the long-chain polyunsaturated omega-3 fatty acids, DHA and EPA, to build and maintain the phospholipid membranes that surround every single one of its 86 billion neurons. These membranes are not merely passive barriers. They are dynamic, fluid, living structures through which every neurotransmitter signal, every electrical impulse, every chemical message must pass. When these membranes are built from the right fatty acids, flexible, fluid, and responsive, neural communication is fast, efficient, and precise. When they are built from the wrong fats, saturated, rigid, and inflexible, neural communication slows, degrades, and fails. The brain is also the most oxidatively stressed organ in the body. Its enormous energy consumption generates massive quantities of reactive oxygen species, free radicals, that continuously attack neurons, mitochondria, and synaptic connections. The brain's natural antioxidant defenses, while sophisticated, are chronically overwhelmed by the demands of modern life: stress, pollution, poor sleep, processed food, and the simple relentless passage of time. The result is oxidative neuronal damage, the molecular foundation of cognitive decline, neurodegenerative disease, and accelerated brain aging. The brain requires a constant, steady supply of specific micronutrients to manufacture its neurotransmitters, the chemical messengers of thought and mood. Serotonin requires tryptophan and B vitamins. Dopamine requires tyrosine and magnesium. Acetylcholine, the neurotransmitter of memory and learning, requires choline and specific fatty acids. GABA, the brain's primary calming neurotransmitter, requires B6 and magnesium. When these nutrients are deficient, neurotransmitter synthesis falters, and the brain's emotional and cognitive performance degrades with it. Nuts provide all of these critical brain nutrients simultaneously: omega-3 fatty acids, powerful antioxidants, B vitamins, magnesium, zinc, selenium, and essential amino acids in a single extraordinarily concentrated whole food package. They are, in the most literal biochemical sense, food specifically designed for the brain. No discussion of nuts and brain health can begin anywhere other than the walnut. It is perhaps the most visually striking example of the ancient doctrine of signatures in all of nature. The outer shell resembles a skull. The inner shell resembles the cerebral cortex with its 2 hemispheres, and the folded nut meat inside uncannily mirrors the brain's characteristic gyri and sulci. Ancient physicians took this resemblance as divine instruction. Modern neuroscience has vindicated their intuition with extraordinary precision. The walnut is the richest plant-based source of alpha-linolenic acid, ALA, the plant form of omega-3 fatty acid. While ALA must be converted to the brain-active forms DHA and EPA, regular walnut consumption has been shown to significantly increase omega-3 index in the brain, improving membrane fluidity and synaptic transmission throughout the neural network. But the walnut's neurological power extends far beyond its omega-3 content. Walnuts contain uniquely high concentrations of polyphenolic compounds, ellagic acid, ellagitannins, catechins, and melatonin that cross the blood-brain barrier and exert direct neuroprotective effects within the brain tissue itself. A landmark study published in the Journal of Alzheimer's Disease demonstrated that walnut extract significantly inhibited the formation and aggregation of amyloid beta plaques, the pathological protein deposits that are the hallmark of Alzheimer's disease, and dissolved existing aggregates in laboratory conditions. This finding has profound implications for Alzheimer's prevention research. The melatonin content of walnuts deserves particular attention. Melatonin is not merely a sleep hormone, it is a powerful brain antioxidant that specifically protects neurons from oxidative damage and plays a critical role in the brain's nocturnal repair and regeneration processes. Walnuts are one of the very few foods that contain bioavailable melatonin in significant concentrations. Clinical studies have shown that walnut consumption measurably increases blood melatonin levels, potentially explaining part of the nut's brain protective effect. Perhaps the most compelling human clinical evidence comes from the WAHA trial, the Walnuts and Healthy Aging study, which demonstrated that daily walnut consumption for 2 years significantly reduced brain aging biomarkers and preserved cognitive function in older adults compared to controls. Brain imaging revealed measurable differences in the structure and connectivity of the walnut consumers' brains, a remarkable finding for a simple dietary intervention.
Almonds hold a unique position in the history of brain nutrition. In Ayurvedic medicine, the ancient Indian medical system dating back 5,000 years, soaked almonds were prescribed as a daily brain tonic for improving memory, concentration, and intellectual performance. Modern neuroscience has now provided the molecular mechanisms that explain this ancient wisdom. Almonds are the single richest food source of alpha-tocopherol vitamin E, the most important fat-soluble antioxidant in the brain. The brain's high fat content and extraordinary oxygen consumption make it uniquely vulnerable to lipid peroxidation, the oxidative destruction of the fatty acids and neuronal membranes. Vitamin E is the brain's primary defense against this process, embedded within neuronal membranes as a molecular shield that intercepts and neutralizes free radicals before they can damage the membrane structure. Studies consistently show that higher blood levels of vitamin E, achievable through regular almond consumption, are associated with significantly reduced risk of cognitive decline and Alzheimer's disease. The famous Rotterdam study, following thousands of elderly participants over years, found that high dietary vitamin E intake was associated with a 43% reduction in dementia risk. A single ounce of almonds, roughly 23 almonds, provides 37% of the daily vitamin E requirement. Almonds are also exceptionally rich in L-carnitine and riboflavin, nutrients that support the mitochondrial energy production pathways in neurons. The brain's neurons are among the most energy-hungry cells in the body, and their mitochondria must function at peak efficiency for optimal cognitive performance. When neuronal mitochondria are well nourished and almonds provide exactly the nutrients they need, neurons fire faster, synaptic transmission is more efficient, and cognitive performance measurably improves. A particularly fascinating study from the SMIL's trial and related nutritional psychiatry research has demonstrated that almond consumption is associated with reduced symptoms of depression and anxiety. The magnesium content of almonds, 1 ounce provides 19% of the daily requirement, plays a critical role here. Magnesium is an essential cofactor for over 300 enzymatic reactions in the brain, including the synthesis of serotonin, dopamine, and GABA. Magnesium deficiency, extraordinarily common in modern populations, is strongly associated with depression, anxiety, insomnia, and cognitive impairment. Almonds are one of the most bioavailable dietary sources of magnesium available.
To truly understand how nuts affect brain health, we must descend to the molecular level and examine what happens inside neurons when the right fatty acids are present versus absent. Every neuron in the brain is surrounded by a cell membrane composed of a phospholipid bilayer, 2 layers of fat molecules arranged tail to tail. The fluidity and flexibility of this membrane is determined almost entirely by the types of fatty acids incorporated into it. Omega-3 fatty acids, abundant in walnuts and present in many other nuts, are highly unsaturated, meaning they have multiple double bonds in their carbon chains that create molecular kinks and bends. These bends prevent the fatty acids from packing together too tightly, keeping the membrane fluid and flexible. This fluidity is not merely a physical property, it has direct and profound functional consequences. Neurotransmitter receptors, the molecular receivers that detect serotonin, dopamine, glutamate, and other chemical signals, are embedded in the neuronal membrane. When the membrane is fluid and flexible, these receptors move freely, adopt their optimal 3-dimensional configurations, and respond to neurotransmitters with high sensitivity. When the membrane is rigid from saturated fat or trans fat incorporation, receptors become sluggish, insensitive, and dysfunctional. The result is impaired neurotransmission, the molecular basis of mood disorders, cognitive sluggishness, and accelerated neurodegeneration. Nuts provide the omega-3 and monounsaturated fatty acids that maintain optimal neuronal membrane fluidity. They simultaneously provide vitamin E and polyphenolic antioxidants that protect these precious membranes from oxidative damage. And they provide the specific micronutrients, magnesium, zinc, B vitamins that neurotransmitter enzymes require to function. In this sense, nuts are not merely brain food, they are the complete molecular toolkit for building and maintaining a high-performance thinking brain. Perhaps the most urgent and relevant dimension of nut-brain research concerns the prevention of age-related cognitive decline and neurodegenerative diseases, conditions that represent one of the greatest medical and social challenges of the 21st century. With Alzheimer's disease alone projected to affect 139 million people globally by 2050, the search for accessible, effective prevention strategies has never been more important. The evidence for nuts as neuroprotective foods is both compelling and rapidly growing. The MIND diet, a hybrid of the Mediterranean and DASH diets specifically designed for brain health, lists nuts as one of its 10 key brain protective food groups, recommending at least 5 servings per week. A landmark study following 923 participants over an average of 4.5 years found that those who most closely followed the MIND diet, including regular nut consumption, had cognitive ages 7.5 years younger than those who least followed it. 7.5 years of cognitive aging prevented by dietary choices alone is an extraordinary finding by any measure. The mechanisms of nut-mediated neuroprotection are multiple and synergistic. The polyphenolic compounds in walnuts, almonds, and pistachios directly inhibit neuroinflammation, the chronic low-grade inflammation of brain tissue that drives the progression of virtually every neurodegenerative disease. These compounds cross the blood-brain barrier and suppress the activation of microglia, the brain's resident immune cells,
preventing them from releasing the inflammatory cytokines that damage neurons. Nuts also protect the brain through their effects on cerebral vascular health, the health of the blood vessels supplying the brain. The brain receives 20% of the body's cardiac output through a delicate network of cerebral arteries and arterioles. When these vessels are damaged by hypertension, atherosclerosis, or oxidative stress, cerebral blood flow is reduced, and with it, the delivery of oxygen and glucose that neurons require to survive and function. The same cardiovascular protective mechanisms through which nuts prevent heart disease, lowering LDL cholesterol, reducing arterial inflammation, improving endothelial function, lowering blood pressure, also directly protect cerebral blood flow and reduce the risk of vascular dementia and stroke-related cognitive impairment. While walnuts and almonds dominate the brain health research literature, the broader nut family offers its own distinctive neurological contributions worthy of exploration. Pistachios have emerged as a particularly fascinating subject of brain research due to their extraordinarily high lutein and zeaxanthin content, carotenoid antioxidants more commonly associated with eye health, but increasingly recognized as critical brain nutrients. The brain, particularly the regions involved in executive function and processing speed, accumulates lutein and zeaxanthin in significant concentrations. A series of studies from the University of Georgia demonstrated that higher brain lutein levels, achievable through regular pistachio consumption, were associated with significantly better cognitive performance, faster processing speed, and greater efficiency of neural networks as measured by EEG and fMRI brain imaging. Pistachios are one of the highest dietary sources of these specific brain carotenoids. Cashews provide an unusually high concentration of zinc, a mineral that plays absolutely critical roles in neurological function. Zinc is essential for the synthesis and release of GABA, the brain's primary inhibitory neurotransmitter responsible for calm focus, anxiety regulation, and sleep quality. Zinc also modulates synaptic plasticity, the ability of synaptic connections to strengthen and weaken in response to experience, which is the molecular foundation of learning and memory. Zinc deficiency is associated with impaired learning, poor memory consolidation, increased anxiety, and accelerated cognitive decline. A single ounce of cashews provides 15% of the daily zinc requirement. Brazil nuts provide the brain with something entirely unique, the single most concentrated dietary source of selenium on Earth. One Brazil nut alone provides approximately 175% of the daily selenium requirement. Selenium is an essential component of the brain's most important antioxidant enzymes, the selenoproteins, including glutathione peroxidase and thioredoxin reductase, that form the brain's primary defense against oxidative damage. Studies have shown that higher selenium status is associated with better cognitive function, reduced risk of depression, and slower cognitive decline in aging. In populations where selenium deficiency is common, supplementation has produced measurable improvements in mood, cognitive speed, and mental clarity. Hazelnuts contribute to brain health through their exceptional proanthocyanidin and vitamin E content, while pecans, the nut with the highest total antioxidant capacity of any tree nut as measured by ORAC score, provide a particularly powerful cocktail of gamma-tocopherol vitamin E and ellagic acid polyphenols that synergistically protect neural tissue from oxidative damage. The emerging field of nutritional psychiatry has produced compelling evidence that regular nut consumption positively affects not only cognitive function, but also emotional well-being, mood stability, and mental health outcomes. This connection operates through multiple sophisticated neurochemical pathways. The tryptophan content of most nuts, particularly almonds and cashews, supports serotonin synthesis. Tryptophan is an essential amino acid precursor from which the brain manufactures serotonin, the neurotransmitter most closely associated with mood stability, emotional resilience, and the prevention of depression. When dietary tryptophan is insufficient, serotonin synthesis is compromised, contributing to depression, anxiety, and emotional dysregulation. Regular nut consumption provides a steady supply of tryptophan, along with the B vitamins and magnesium required by the enzymatic pathway that converts tryptophan to serotonin. Magnesium, abundant in almonds, cashews, and Brazil nuts, plays a particularly profound role in anxiety regulation. Magnesium functions as a natural NMDA receptor antagonist, blocking the overactivation of glutamate receptors that produces the hyperexcited neural state experienced as anxiety. Multiple clinical trials have demonstrated that magnesium supplementation reduces symptoms of anxiety and depression in deficient populations. Given that an estimated 50 to 60% of adults in developed countries consume less than the recommended daily amount of magnesium, nuts represent a critically important dietary source of this brain-calming mineral. A large-scale epidemiological analysis of the NHANES dataset found that adults who regularly consumed nuts had significantly lower rates of depression, anxiety disorders, and poor sleep quality compared to non-nut consumers, a finding that remained statistically significant after controlling for all other lifestyle and socioeconomic factors. The authors concluded that the combination of omega-3 fatty acids, magnesium, tryptophan, vitamin E, and polyphenols in nuts created a synergistic neuropsychiatric protective effect that no single nutrient could replicate alone. The scientific evidence is clear and consistent. Regular nut consumption is one of the most powerful, accessible, and evidence-based strategies available for protecting and enhancing brain health across the entire lifespan. But how should this evidence be translated into practical daily nutrition? The optimal daily serving for brain health, supported by the preponderance of clinical evidence, is approximately 28 to 30 grams of mixed nuts, roughly a small handful or 1 ounce. This modest quantity, consumed daily, has been shown to produce measurable improvements in cognitive performance, mood, and brain aging biomarkers within as little as 8 to 12 weeks of consistent consumption. For maximum neurological benefit, variety is essential. Different nuts provide different brain-critical nutrients: walnuts for omega-3 and polyphenols, almonds for vitamin E and magnesium, pistachios for lutein and zeaxanthin, cashews for zinc, Brazil nuts for selenium. A mixed daily handful that includes all of these nut types provides the brain with its complete nutritional needs in a single convenient serving. Timing matters as well. Consuming nuts with breakfast or as a mid-morning snack takes advantage of their slow-release energy profile, providing the brain with a steady supply of glucose and fatty acids throughout the cognitively demanding morning hours. Students, knowledge workers, and anyone requiring sustained mental performance throughout the day will particularly benefit from this morning nut consumption strategy. Raw or dry roasted nuts are preferable to oil roasted or salted varieties. Commercial roasting oils often add omega-6 fatty acids that compete with the brain beneficial omega-3 content, and excessive sodium can blunt the cardiovascular benefits that indirectly support cerebral blood flow. The evidence assembled by decades of nutritional neuroscience research leads to one clear, simple, and actionable conclusion. Eating a small handful of mixed nuts every single day is one of the most powerful things a human being can do to protect, nourish, and optimize the health of their brain. From the walnuts' omega-3 fatty acids rebuilding neuronal membranes to the almonds' vitamin E shielding those membranes from oxidative destruction. From the pistachios' lutein accelerating neural processing speed to the Brazil nuts' selenium powering the brain's master antioxidant enzymes. From the cashew's zinc sculpting synaptic plasticity to the pecan's polyphenols extinguishing the neuroinflammatory fires of cognitive aging. Each nut contributes its unique molecular gifts to the brain's health, and together they form a complete, synergistic, and extraordinarily powerful neuroprotective package. The ancient physicians who prescribed almonds for memory, the traditional cultures who revered walnuts as brain food, the grandmothers across every culture who pressed handfuls of nuts into children's palms before school, they knew something profound that modern neuroscience is only now fully explaining at the molecular level. The brain knows what it needs. It built a food that looks exactly like itself to remind us. Eat your nuts, feed your brain. The evidence has never been clearer or more delicious.